Hello guys, today we are going to discuss about physiology of muscle contraction. Okay, so before directly jumping to the topic, I want to tell you about some terms and their definitions. Like, first of all, filaments, and we also call them as microfilaments. Okay, microfilaments. What are these? These are structural and functional units of sarcomere. Sarcomere. What is the sarcomere? Sarcomere. Sarcomere. This is the area between two adjacent Z lines. Okay, this is the area between two Z lines, and it is also the unit of muscle contraction. What? Unit of muscle contraction. Okay, now you will ask what are these Z lines? We are just going to discuss about them. Okay, so first of all, I want to tell you about these filaments. These filaments are of two types: thin filaments and thick filament. Okay, now these thin filaments are formed of protein, actin, tropomyosin and troponin. These are formed of protein, actin, tropomyosin and troponin. Okay, these thick filaments are formed of protein, myosin. Okay, so now I want to tell you what are these thin and thick filaments. Okay, now from here I want to tell you, see, this line, this thing is this below and above thing, this. Okay, this is actin. Okay, what? That actin, tropomyosin and troponin. These are thin filaments. Okay, they are made up of actin, tropomyosin and troponin. Okay, this red one in between. This is myosin. What is myosin? Myosin is thick filament. Okay, this in between red one. So here what will happen? Now, in between of these two Z lines, these are two Z lines. Okay, these are two Z lines. Okay, there are so many Z lines like this. Like, see, one Z line, then another Z line, then another Z lines, and they have like these structures. Like, this is that actin or thin filaments. Okay, now in between them we have this myosin. Okay, in between we have myosin. Okay, there are so many structures like this, and here what happens? This myosin have some myosin heads. As you can see, this blue, these are myosin head. Okay, myosin head. They attaches to these thin filaments, and causes muscle contraction. Actually, what happened? These myosin heads will attach to this thin filament. Okay, they will attach to this thin filament whenever we wanted contraction. Okay, and what happens when they attach to this thin filament? Okay, these myosin heads, they will pull that actin towards each other. So they will pull the actin. Okay, so there is one question, like, which protein will move? Obviously these actin will move, or this thin filament. Okay, this thin filament will move and they will come close to each other. Okay, what will happen? This will move and they will come close to each other. Okay, I hope you are getting my point. They will move like this and they will come close to each other, and here the contraction will happen in the muscles. Okay, now this thing is clear. So I also told you that this thin filament, or we also called it as actin, because see, this thin filament is mostly made up of actin. Okay, so we also call it as actin filament. Okay, so this actin filament or this thin filament will come close to each other and muscle contract, but myosin will not move. Myosin will always remain at its place, at its own place. Okay, these myosin heads will join and they will pull these actin filament each other, and there are so many structures like this in a cell, as I told you, and because of which your muscle will contract means it will come close to each other. Okay, now After this, I want to tell you about some bands or zones in this. As I already told you, in between or in middle, like this in middle part, we have line that is known as M line. Okay, this M line, okay, is in between of this myosin, okay, this myosin, or we can also say it is in the middle of two Z lines. Okay, it is in middle of this M line. M line is in the middle of two Z lines. Okay, two Z lines. These are Z lines, as I told you. It is in between or in middle of this. Okay, or we can also say it is in the middle of myosin filament or thick filament. Okay, now there are some more bands. One more thing. Here we have this yellow thing is calcium. What? Calcium. We are just going to discuss about their mechanism, like how this head bind to here, okay, to actin filament, how this myosin head will bind. Okay, before that, let me tell you something about these bands. There are some bands in this. See, this myosin head or this myosin ending is joined by this Z line by titin, Tin. Okay, titin. So this titin is joining this myosin ending with Z line. Okay, and we call this part as I band. What? I band. Okay, here also the same thing. Okay, so here also we have this I band. Okay, now the distance between these two endings is known as H zone. Okay, it is a distance between these two actin filaments. Okay, this is H zone. Okay, now the distance of this myosin filament or this thick filament, this red one, is known as A band. What? A band. A band. Okay, this thing in between, in middle. Okay, this is known as A band. These bands are clear. In between we have M line, then H zone, A band, I band, and Z lines. And the distance between two Z lines is known as, we already discussed, is known as sarcomere. Sarcomere. Okay, the distance between two Z lines. Okay, as we discuss about here, see, this sarcomere, this is the area between two adjacent Z lines. And it is also the unit of muscle contraction. Okay, I hope this thing is clear for you. During muscle contraction, if someone wants to ask you what will happen, see, when the muscle will contract, these two Z lines will come close to each other. Okay, so you can write this as a summary thing. Okay, so see, you can pause the video and write these things also, but before pausing, understand this thing. H zone is the distance between two actin filaments. Okay, I band is the distance between Z lines adjacent, is the distance between Z lines and myosin filament. Okay, distance between Z line and myosin, as I already told you, these are like Z lines and Z lines and this thing. Okay, this titin part is I band. Okay, now A band is the length of myosin filament in one sarcomere. Okay, sarcomere is what? Is the distance between two adjacent Z lines. Okay, so it is distance between two Z lines is known as sarcomere. Now during contraction, what will happen? The H zone and I band size will reduce or they almost disappear. Okay, how? H and I band, see, this is H zone. Okay, this is I band. Okay, you know, this is I band. Okay, this is I band. Now what is happening when they will come close, like see, Z line will come close, I will use Z line for that, these will come close to each other. So what will happen? See, the actin filament is, okay, for your understanding. Okay, see, these Z lines will come close to each other. So when they will come close to each other, what will happen? This A band will also, this H zone will also come close to each other. Okay, this actin protein will move. So if someone will ask you which protein will move, actin. Okay, this actin filament or thin filament will move. Okay, because myosin head will pull them towards each other. Now just imagine this is the M line or middle line, M line. So what will happen to that I band, this I band? This will reduce so much that it almost disappear. Okay, what will happen to this H zone? You know, this is H zone, no? This H zone, this is H zone. it's also almost disappear. Okay, and what happens to this sarcomere? Sarcomere is what? Distance between two Z lines. It will also reduce. Okay, but it not disappear. Okay. So see, H zone and I band size reduce, almost disappear. A band will remain same. This is that myosin filament thing. Sarcomere sizes reduce. Sarcomere, the distance between two Z lines, okay, will also reduce during contraction. Okay, this thing is clear regarding bands and zones. Now, I want to tell you how these attach, like myosin head will attach to this thing. Okay, so see, imagine this is the cell, any cell membrane. Okay, this is that cell membrane, and here some potential arises. Okay, some potential, action potential arises, and that action potential will irritate potential dependent calcium channels. It will irritate potential dependent calcium channel, and they are also known as AKA. They are also known as voltage gated channels. Okay, this thing is clear. These are also known as voltage gated channels, and these voltage gated channels are present in T tubules. What? T tubules. These T tubules. What are T tubules? These are the structures like this. This U-shaped structure is T tubules. Okay, and they are filled with calcium. See, between two T tubules, see, this is one T tubule. Okay, this is another T tubule border. In between them, we have longitudinal tubes. Okay, longitudinal tubes, and they are filled with calcium. They are filled with calcium. Okay, so whenever there is irritation, like action potential arises, it will irritate these potential dependent calcium channels or voltage gated channels. Okay, so they will open and remove calcium outside, and the calcium concentration will increase a lot. Okay, now after the calcium concentration will increase outside, I want to tell you, as you studied in biochemistry, or you will study in biochemistry, we have some active sites. Those active sites are specific for some molecules. Okay, they are specific for some molecules. So these actin filament, see, these actin filaments look like this. This is M line, that middle line, or this is Z lines. We are just zooming in inside this. See, okay, this is myosin. This is that titin. Okay, that I band thing, as we study I band. Okay, this actin filament have this this blue thing is known as tropomyosin. What? Tropomyosin. Okay, and this tropomyosin have some active sites. You know, active sites are specific for some molecules. Okay, and these active sites are specific for this protein, and this is known as troponin. Okay, now this troponin, see, this troponin, and as we already discussed that here calcium will increase. Okay, and the troponin loves more calcium than this tropomyosin site. The troponin is like, I am always attaching with this tropomyosin, let's try something new. So troponin loves more calcium. Okay, so the troponin will bind with calcium and leave the active site of tropomyosin, okay, active site of tropomyosin. And now this active site is open. Just imagine from here the troponin is removed. Okay, because calcium takes that. Okay, so here on this myosin head we have ATP. Okay, now on this myosin head the ATP will hydrolyze. Okay, and it will divide into ADP plus inorganic phosphate. Okay, now this inorganic phosphate will be removed from here. Okay, now it only have ADP, and it will go and bind above here, where it will go and bind with upside with tropomyosin active site. Okay, so myosin head also have some active site. That active site normally have ATP. Okay, so during contraction, that ATP hydrolysis means it break down into ADP plus inorganic phosphate. Then this inorganic phosphate will be removed, and we only have ADP. And when it have ADP, it will go and bind with the active site of tropomyosin. Okay, it will go and bind with the active site of tropomyosin. This thing is clear. As I already told you, from T tubules we remove calcium. That calcium takes this troponin, okay, with it. So when this troponin will be removed, the tropomyosin active site is open, and when its active site is open, the myosin head will come and attaches to that active site. Okay, and when it will attach to these active sites, they will pull. Pull means they will pull them like this. Okay, they will pull them like this. So the contraction happens. As I already told you, these Z lines will come close. Okay, so these myosin head will join here and they will pull these Z lines. They will pull the actin filament and Z lines will come close to each other. Okay, and the muscle contract. Okay, in between we have myosin. The muscle contract. This is a simple thing. Okay, this is M line, this is Z line. Remember this thing. Here we are only discussing the half part of this. Like, see, this is M line and this is Z line. Okay, now I hope what I teach you is clear till here. Okay, the same diagram, this diagram is same with this diagram. So this diagram is also same. As you can see, this is M line, this is Z line. We have this actin, this B, okay, this actin thing, okay, actin protein. This actin protein have tropomyosin molecule over it. It's like binding over it, okay, like a rope, you know, rope. Okay, now on this tropomyosin we have active sites, and those active sites are covered with troponin. These are covered with troponin. Okay, these are myosin, as I told you, myosin. These are myosin's active head, these are myosin's head. Okay, they also have active site over here. We already told you they have ATP. Okay, during contraction the ATP will hydrolyze into ADP plus inorganic phosphate, and this troponin will go with calcium. It will bind with calcium and leave the active site of tropomyosin, and hence this active site is open. And below also there is myosin head only have now ADP. One inorganic phosphate is removed, so it will go and bind above. Okay, above where? On the active site of tropomyosin. Sorry, I am taking too much time, but it's for you, for a better understanding. Okay, for better concept building. Okay, Z line, M line, myosin, calcium and troponin. Now here also this diagram is also the same thing in a systemic manner. Okay, first of all, see, here we have ATP. This ATP will break into ADP plus inorganic phosphate. Okay, when this will break into ADP plus inorganic phosphate, this IP is inorganic phosphate, then this inorganic phosphate will be removed. You can also write it down, just these points, 4, 5, 1, 3. Okay, now first of all I want to explain you this thing, then write as per your thinking. Okay, this inorganic phosphate is removed, and see here ADP is left. Okay, and then this myosin head will go and bind above. Okay, with tropomyosin active site. Bind to where? Bind to tropomyosin. AS means active site. Okay, and then they will pull actin filaments like this. Okay, and we call this process of pulling as power stroke. Okay, now actin gets pulled toward middle of sarcomere. What is that middle of sarcomere? M line. Okay, actin gets pulled towards middle of sarcomere, as we call it as power stroke. Now what will happen? This ADP will be removed because of this power stroke. Okay, this ADP will be removed, and when it will not have any ADP, ADP, so a new ATP will come and bind here, and when it will bind here, the myosin head will remove or will leave the tropomyosin active site. Okay, ATP, we can write also here, ATP join and myosin leave active site of tropomyosin. That's why we call it as tropomyosin. Okay, so ATP join and myosin leaves active site of tropomyosin. Okay, and then again the active site will open. Okay, then again that troponin will be like, now I am bored with this calcium, then it will leave the calcium and come back again and join here. Okay, this troponin or new troponin will be formed, or it will come and join here. Okay, then the same cycles goes in a cycle manner. Okay, round and round. See, then again ATP hydrolyze. Okay, after hydrolyze it will break into ADP and inorganic phosphate. Then the same cycle goes on and your muscles contract. Okay, now let's all of this thing, whatever I told you, in a summary part and in a systemic manner form. Okay, so first of all, what happens? Action potential is generated, as I already told you. action potential generated, and it will reach T tubules. Okay, now on this T tubules, calcium channel will open. What are this calcium channel? Voltage gated calcium channel. Okay, will open. Now endocellular calcium increases. Okay, now endocellular calcium increases. Okay, from that longitudinal tubes the calcium will come out. Okay, and it will increase the endocellular calcium level. Okay, so calcium will bind to troponin. Okay, calcium will bind to troponin. Because of this, what will happen? This leads to open active site of tropomyosin. Okay, now active site is open, and at the same time, as I already told you, on myosin head, ATP will hydrolyze or break into ADP plus inorganic phosphate. Okay, now this inorganic phosphate will be released. Okay, and cross bridge form. What is cross bridge? I think I forgot to tell you. When this myosin head will join to this, when this thin filament or this thick filament will join together, okay, by this myosin head, is known as bridge formation. Okay, I hope you get my point. Okay, whenever they will join by this myosin head, so this is known as cross bridge formation between thin filament and thick filament by myosin head or myosin protein. Okay, now will be released this inorganic phosphate, okay, and form cross bridge. Okay, now and myosin will attach to active site of tropomyosin. Okay, and it is known as cross bridge. Okay, now myosin will leave ADP, or myosin head will leave ADP. Okay, then what will happen? A new ATP will come. This a new ATP, okay, will come and attach on myosin head. Okay, come and attach on myosin head. Okay, because of which it leads to, see, when that ATP will join, suddenly the cross bridge formation will be over. Okay, so it leads to unbinding of myosin and thin filament. Okay, what is this thin filament? That actin filament. Okay, unbinding of myosin and thin filament. Okay, and this is known as cross bridge formation ended or over. Okay, cross bridge formation is over, okay, or ended. So now there is no cross bridge. Okay, so this is the kind of summary thing you must have to write about this. Okay, of what all I told you. If you want, you can draw these diagrams too, if you want. Okay, this one and this one, this cycle, this diagram, okay, and this starting diagram. If you want, you can draw it. Okay, you also have to write this thing, this filaments part. Write this filament part. You must have to write about this filament part. Okay, then you must have to write about this summary thing. Okay, then you must have to write this summary also. this is the summary of that bends. Okay. And if you want, you can draw this diagram also.